blue. Now, people online can hear us, and that's where you'll hear them from as well.
Okay.
Okay, they won't come up through the room, they'll come up through the sky.
Okay, so if I ask them a question, it'll come through.
Exactly. You'll talk directly into that.
Okay.
So welcome everybody. We're going to get ourselves underway for today's Health Research Institute seminar series. My name is Margot Parks. I know most of you, but I'm in the School of Health Sciences and one of the team, the fabulous team of people who pull these seminar series together as an opportunity to celebrate the quality and excitement of health research here at UNBC. So just a little bit about the HRI seminar series. We draw on contributions from across campus because there is this vibrant health research culture across UNBC. And where our emphasis has really committedly been in our first few years to make sure that we're profiling researchers here. And we've still got a long way to go just on getting through the fabulous researchers at UNBC. So those of you that might not have presented yet of our fabulous colleagues know that there will be shoulders tapping shortly. Or if you've got out of it last time, there's still more opportunities. So we see this as a real team effort that's also got a range of sponsors and others. And so we are having a rotating, we try to move across the different sort of themes and scope across our whole group. And on March the 12th, we have a sudden vacancy. So for those fine colleagues that are here that are burning to be able to present and share with your colleagues an aspect of your work, importantly, it's really tough to find the right time of day, time of week to host these events, but they are recorded and we do get a lot of traffic, people being able to see these videos elsewhere. So it's also a nice way to have a documented talk that can be shared with other colleagues and as part of us sort of celebrating the work beyond the actual day. So we sometimes have people logging in and then also logging in at a distance. So March 12th, awaiting your hands up volunteer. So please let us know after today's talk if that's of interest to you. And thanks to Bree, and who's the administration assistant for the Health Research Institute. We maintain and communicate and get the word out. And we also have a great team of those of us that are here. We've got Chelsea and Sarah and myself, and Rachel Wells, along with our student reps, some of who are frantically finishing their thesis right now. And we also try to time these events with when there are research seminar classes, but this semester we don't have a research seminar class at the same time, so we're always juggling. And importantly, we're always open to your suggestions. So particularly it's great to see some faculty from that have come to listen to today's fabulous talk, and we're welcome to how you think this might, we can continue to do this and keep a thriving celebration of what's happening here at UNBC. And so speaking of that, it's a very great pleasure for me today. It's always, I think these are actually very difficult presentations to do to our colleagues, our proximal colleagues. So I haven't had to do it yet, but Sarah fortunately put her hand up for this month.
I did, yeah.
And it's a really nice moment, in fact, to hear our colleagues present in a relatively intimate way, just, this, often even with a small audience, to just share with people what you're doing and to give them a glimpse of the work you're doing. So it's a real treat for me today to have that chance to listen to Sarah's talk, which is about biological determinants of obesity and diabetes, the biomedical perspective to disease prevention and management. But it's also a really nice moment to actually introduce Sarah as a really valued colleague in the Health Research Institute, and as a leadership council member, as somebody who's really been doing a fantastic job at helping to figure out how we can create connections between and across the fabulous researchers at the NMP and other people at other parts of campus. And so it is actually, we spend a lot of time talking about those big picture questions. So it's a real treat today to get to hear Sarah present about this work. I think it's really worth mentioning that some colleagues have to navigate particular dynamics to progress their research, and I'm always in admiration of people that are cutting across NSERC and CIHR and navigating a whole lot of really innovative ways to pull together the exciting portfolio of research that we're going to hear about. Importantly, Sarah and I also share the curious role of being Canada Research Chairs, which is a privilege, but also a particular story in itself. So it's really nice to introduce Sarah as a Canada Research Chair at the Northern Medical Program, who's going to dive in. I'm not going to try to capture her work because I'm going to hand that over to Sarah. But a big thank you, Sarah, for all that you do to make these seminar series possible and the Leadership Council, etc.
Thank you. Very nice.
I'll hand over.
Okay, can everyone hear me? Is that okay? I'm just going to stand up because if I sit, that light is directly in my eyes. So if there is anyone on the line, I hope you can hear me. And please, can they ask a question anytime?
Freeze monitor.
Okay, great. And just please feel free to interject anytime, and that's for the audience here too. In my perfect type A form, I have prepared slides and a talk, but please feel free to interrupt me at any moment. So thanks, Margot, for that introduction. I actually did find it quite challenging to make the talk for this session today, knowing that we have quite a general audience within this seminar series. I have been part of organizing these seminars, and one of the goals has been to have to cross disciplines across health research. And so today I will be bringing a biomedical perspective, and I'm actually going to subject you to the work that we've been doing, the most foundational work that we've been doing in the context of diabetes and obesity, which is NSERC. So I wanted to give myself that challenge because I think it's really important to share with people the breadth of health research. And so I went right back to some of our foundational work. So what do I do here? Okay.
Pull up the PowerPoint at the bottom.
Okay.
And then switch it to yours.
Got it.
And then there'll be...
Perfect. Okay, great. All right, so I'll just start by saying that I have no conflicts of interest to disclose. And really the goal for me today was to introduce you to the area of health research that I'm very passionate about, which is the endocrine or hormonal regulation of energy metabolism as it relates to diabetes and obesity. And so I'm going to start my talk really highlighting some of the key biomedical findings that have happened historically that have really transformed the way we think about metabolic disease and the way we treat metabolic disease. And then the second half of my talk, I'm going to introduce you or share with you some of the work that we're doing here at UNBC to try to advance that field. And obviously the goal of our work, like all health research, is to improve the health of patients. And so how our work ties into hopefully improving prevention and treatment for diabetes and obesity. So that's sort of the goals, and you can let me know afterwards if I was successful in doing that. I'm going to apologize on behalf of my research group who are not here today because many of them had conflicts with their classes during this time frame, which is unfortunate, but I'm going to showcase some of their work, and this is definitely a team effort and not something that I do on my own. One other piece before I get going, I do have two images in my slides of children with endocrine disorders, which can be a little bit shocking when you first see the picture, so I'll try to warn you when those come up, and in the next slide I have one here. So it's very well established that hormones play a role in regulating metabolism. And this picture here is a picture taken in 1922 of a child who has diabetes. Diabetes is an example of a disease where a hormone is lacking or hormone action is lacking, and the result is metabolic decompensation. And so does anybody know the significance of the year 1922? No? Yeah, thank you. There you go. Good Canadian, or not. She's like, you're good Canadian. Are you Canadian now? Awesome. Okay, so you are a very good Canadian. Was that on your test? Was that on your test about? Was it on there?
It was there. I can't remember.
Okay, got it. So it might have been on your test for Canada. This is Canadian history in the making. So in 1922, a group of scientists at the University of Toronto were doing some lab-based experiments where they found a way to isolate extracts of the pancreas that maintained the hormone at what we now today know to be insulin. And very quickly, they were able to move that discovery from the lab to the clinic and treat patients who were in this state, and they were able to reverse the symptoms of diabetes. So this is a picture of the same girl. This woman's name is Elizabeth Hughes. And you can see very shortly after she received those extracts, which now contained insulin, the metabolic phenotype was reversed. And so this was a really miraculous biomedical finding that was awarded the Nobel Prize in 1923. And so this is rather an extreme example, but obviously very close to my heart, and really some of the stories like this that has inspired me to work in this area of science. So this was an example of a very rapid translation of biomedical knowledge to the clinical field. So I just put this slide up here just to make sure we're on the same page when I keep using the word biomedical research, the area of science devoted to studying the most fundamental processes of life, with the goal that we can translate that knowledge to the prevention and treatment of disease, and also to understand the genetic and environmental, and I'm going to add in that social factors that lead to the presentation of disease or that dictate health. And so this is sort of the angle that we're coming from, and there's a few biomedical scientists in the room as well, so they'll appreciate that slide. So biomedical research is just one piece of the health research continuum that the knowledge, generally speaking, the knowledge that comes from the biomedical domain needs to be integrated with information that's developed in other domains in order to improve health. And I think that's the goal of all health researchers, to improve health. So today I'm going to be really focused in this biomedical domain and in the area that one area that I'm extremely passionate about, which is in the prevention and treatment of obesity. But of course, the pieces that I'm going to discover in my lab are not going to solve that problem. It's going to require information integrated from across the health research spectrum. And that's because like many chronic diseases that are affecting our society today, things like cardiovascular disease, diabetes, dyslipidemia, mental illness, these are very complex, multifactorial diseases that are influenced by many factors, so physiological, psychological, environmental, socioeconomic, as well as genetic factors. And so I just made this little slide just to sort of simplify, and I apologize for people who are in public health, they might be appalled by this slide, but I sort of think of them as the inside factors and the outside factors that are sort of influencing our health. And I've just listed a few here. So some of the outside factors might be this this gentleman here has chosen to live in this lovely looking city. He's moved, like many people, to an urban environment, and he maybe has an office job, and this will affect how sedentary or active he can be in his life. He will be surrounded by an excess availability of unhealthy foods, and actually challenged by a high cost of actually choosing to eat in a healthy way. Depending how or where he lives within the city, it might change how much he uses his car to move around rather than walking. And even the city planning will affect his day-to-day lifestyle in terms of his choices. As well, we have inside factors within this individual that are going to mean that he responds differently to his environment. So his genetic predisposition to his sort of predetermined body weight or efficiency, for example, within the way we use our nutrients, and also his physiological response to things like stressors. For example, stress is known in some individuals to increase appetite, or to his mental wellness. And also don't forget the microbiome. I put that in there because that's probably one of the newest areas of research, or one of the new areas of research in obesity, where we know that the nutrients that we consume affect the microbiome within the gut, and that has direct effects on how we regulate our food intake and also our activity. So just to say basically that I don't want to be up here as the lab-based scientist saying that we're going to come up with a magic bullet to obesity, that really we are one part of a big puzzle, and that's why things like the HRI are extremely important to me. And one of the advantages I've had at working at UNBC, I used to work on the 5th floor as a postdoc of the cellular and physiological sciences, surrounded by diabetes researchers. Actually, they all worked on islets. That's how specific it was. And when I was in Cambridge, same thing. I worked with people who were all working on insulin resistance. And actually, one of the values of being at UNBC is we get to come to seminars where I will hear people talk about the outside factors and other inside factors that I maybe hadn't thought about. So all this to say that now I'm going to take that complexity and boil it down to a really quite simple energy expenditure equation when it comes to obesity. And that is, we think about it as if we have the amount of energy that's coming into the system through feeding equals that of the energy that's being expended by processes like basal metabolic rate, thermoregulation, or physical activity, then body weight should remain stable. However, we know that all of those factors are influencing this energy balance equation, so it is actually quite complex. And we can see I'm using obesity as my example here because, of course, that's the focus of my talk today. But obesity then represents a state of dysregulated energy balance. And in our field, we refer to that as a chronic state of positive energy balance, where that's just a fancy way of saying that the in is outweighing the out. Okay. So I now want to, in my first sort of little slide about diabetes, I highlighted a hormone that regulates metabolism. I think that we're all familiar with. But actually, hormones are also being shown to be critical drivers of this energy balance equation. And I think many of you are familiar with that now, but that's only knowledge that's about 20 years old, 25 years old. And so I want to introduce you to one of those hormones, and I think you guys will probably guess which hormone I've decided to pick as my star of that show. But before we go there, I just want to make sure we're all on the same page and we all understand what a hormone is. So can anyone tell me what a hormone is? Don't worry, I won't make you do it like your biochemistry classes, physiology classes. What does a hormone do in the body? Anybody? Okay, that's all right. It's like, have you ever heard chemical messenger? So it carries messages, right, between one place and another in the body. So good, you guys are getting your basic physiology lesson. I feel like I've helped you to have a dinner conversation tonight. So hormones are chemical substances, and I said chemical substances because they're molecules. They can be proteins, they can be lipid derivatives, they can be derivatives of amino acids, but they're produced and secreted by a cell, and this is a very classic pathway that I've put here. They are secreted by that cell, and then they usually travel for a distance in the body, often through the blood, and then they act on a target cell right here. And the way that they have that specificity to act on a cell is you have this receptor that's waiting to sort of catch that hormone. And so that lock and key analogy that people will use in basic biology courses is that's how hormones get their specificity, even though they're sent out into the abyss. So that's what I mean when I refer to hormone, because I'm going to talk about hormones a lot today. So as I mentioned, hormones do regulate energy balance. And again, I'm going to have a picture here of a child with dysregulated energy balance. So just to warn you here, this is a picture of a child who whose energy balance is dysregulated. He is in a chronic state of positive energy balance due to a very severe hyperphagia and a decrease in energy expenditure. And what's interesting about this particular case is that his obesity is due entirely to a single gene mutation. Okay, so none of the outside factors are at play here. This is about a genetic mutation that is causing a hyperphagia. And I've put this little picture of this mouse because he has the same gene mutation. Does anybody know what the gene is that I'm referring to that's mutated? Starts with an L? Leptin. There you go. That's great. So leptin is the gene that is mutated in both this mouse and this child here. And this guy is actually, this is a naturally occurring mutation, and they didn't know what the gene was, and that's how he got his name, the obese mouse. He was the obese mouse. And in 1994, Jeffrey Friedman at the Rockefeller in New York identified that the gene that was missing was leptin. That only happened in 1994. And then later in 2004, actually it happened, Sadaf Farooqi and Steve O'Reilly in England identified a family from Pakistan who had the same mutation. So it's a very, very rare condition. We're not talking about generalized obesity here, but what was so significant about this discovery is that it identified that there were inside factors regulating body weight. And to this day, we are aware that there's a lot of stigma associated with obesity that says it's everything that you're doing to yourself, it's everything what you're choosing to do. But actually, it's a multifactorial chronic disease, like other diseases we see in our society today, like hypertension, diabetes, dyslipidemia, et cetera, that have both outside and inside factors influencing. So leptin then, you're going to get a little lesson because you're saying, but how does this happen with just leptin? So leptin really opened up this whole world of endocrine regulation of energy balance. And leptin is an example of a sensor in the body that tells the brain how much nutrients do we have stored. So in times of famine, and I'm going to do it this way around, when you have, say, low amounts of adipose tissue, you will produce leptin proportionate to the amount of adipose tissue you have. So if you have low amounts of adipose tissue, you'll create low amounts of leptin, and that tells the brain, we better increase activities for food-seeking behavior, and we are going to decrease those expensive activities for energy, like reproduction, for example. So I don't know if you've ever heard high-level athletes that have low amounts of adipose tissue who do not go into menstruation. That's because they have low levels of leptin, which are preventing the activation of that reproductive axis. Okay. And then the opposite is sort of true. And if someone asks me a question, I'll answer why sort of. If you have high levels or large amounts of adipose tissue, then you have high circulating levels of leptin, and this will, in theory, decrease the amount of energy spent on food-seeking behavior and increase time for things like reproduction, energy expensive things like reproduction. So that's an example of how the body is very clever. It communicates from the adipose tissue to the brain. And prior to that, we really just thought adipose tissue was big sink to put your excess calories. But now we know it's an endocrine organ actually communicating with other parts of the body to maintain this energy balance equation. And just to close the loop, this child here was a candidate for leptin therapy because they knew what the mutation was. And so you can see this little guy's obesity was resolved shortly after starting leptin therapy. He is a little bit of short stature for his age because leptin has effects on the growth axis, but he was able to resolve his obesity. Yep.
You mentioned that there's a leptin receptor, or is the receptor called leptin and the hormone called leptin?
The leptin receptor is called leptin receptor. Yeah. Often we do that, not always. Usually we have a nice little pairing happen. So this is a very, again, the examples that I've given you are very extreme, and obviously generalized obesity in the population is not related to a mutation in the leptin gene, but it did have a significant implication about how we think about metabolic disease and our approach to it. And so since the discovery of leptin, there have been hundreds of hormones, or maybe tens of hormones, that have been identified to be involved in balancing this energy balance equation. Some are long-term signals, like leptin coming from the adipose tissue communicating with the brain about nutrient status. Others are short-term signals coming from the gut. Oh, I feel full, turn off my desire to eat. And so we have a lot of communication happening from the periphery back to the brain, and then from the brain back out to control our intake and also our energy expenditure. Okay, so now we're going to get to, maybe I'll just pause there and see if there's any questions before I continue. That's your history lesson. I think I'm done with the history lesson. So, okay, we're good. All right. So this is where my lab has really been interested, one area of interest in my lab for the last, almost 10 years, I guess, has been we've identified a hormone that called PACAP, which probably none of you have heard of, and I'll tell you a little bit about in a couple of slides here, that seems to be involved in regulating this energy balance equation. And the information that we're finding is we think that it's regulating this energy expenditure side of the energy balance equation. And so this is sort of quite foundational work and most of this work has been funded by NSERC.
Sarah,
yeah.
Just to check, your little red arrow column is body weight.
Yeah. It is, yeah. Which is sort of, it's sort of correct. I have this. I've just kept it here. When you compare the two diagrams, it's correct. So I sort of say stable or it gets larger, but yeah. Okay, so I thought this would be a good time to introduce to you my lab and the people that are doing the work. So these are two pictures that we've had from our annual hiking trip. That is now annual. It's happened 2 years in a row. Nice. And so, so we are going to try to continue with that. And I've listed my current lab members, and I also have listed some past members, particularly Megan, Eric, and Tech, who have contributed to the work that I'm going to show you today. And then we have these 4 members here who have just joined the lab since September. So, and this is I was asked about a week ago, so with Margot, to have a one-page, one-slide thing for the NMP to introduce students to the type of research that you're doing. So I developed this slide. And so I just thought I would include it to just let people know that sort of this is a slide that represents my program of research here at UNBC. I think the long-term, I'm trained in this domain here, the biomedical domain in the area of endocrinology, obesity, and diabetes. But I think that the goal for my long-term career is that I make sure I have a research program that sort of tries to work towards translation of the biomedical knowledge that we're identifying in the lab to other areas, particularly into perhaps the clinical domain. So right now, while the majority of my work is still in the area of biomedical research, we do have a clinical project where then Larstone is sitting up there, and she heads that project, looking at cardiometabolic risk factors in people with mental illness. And we also do have a collaboration with pharmaceutical sciences at UBC, where we're looking at, with our expertise in adipose tissue physiology, we're helping a lab that is interested in developing drug delivery tools. And so we're trying to find novel ways to deliver drugs to adipose tissue. So that's sort of my whole program of research. I was very tempted to talk about this here to this audience here, which I felt might be a little bit closer to some of the other health research that's going on, but I chose to make it a challenge and go for something a little bit more hardcore biomedical. So that's what I'm going to share with you today. Okay. So as I mentioned in the previous slide, this is the framework which I want you to be thinking in and what's sort of guiding our research. And so we're looking here at this side of the energy balance equation. So I'm going to introduce you to this hormone that I say, unfortunately, I've been working on for too many years of my life. I worked on this hormone during my PhD, and I kind of vowed I would never work on it again because it's not sort of straightforward. But then my supervisor retired, and it was an interesting story, so I ended up working with PACAP again. So I feel like it's a life sentence for me. So what is PACAP? And I actually wish Annie was here because Annie might be the only person at UNBC really who knows what PACAP is. So if it's a stress hormone.
We do now.
Yeah, now you do know. And that's why I call it PACAP, because this is its name here. So PACAP is a neuropeptide that was discovered in 1989. And over the last few decades, or 30 years, I guess now, it's involved in all sorts of different processes. But really what started to come out about what this peptide does is a very high-level regulator of the stress response. And it's a regulator of the two key stress axes in the body. First, we have the HPA axis, or the hypothalamic pituitary adrenal axis, and you might be more familiar with the hormone that's produced from that axis, which is cortisol. So it regulates the secretion of cortisol from the adrenal cortex in response to psychogenic stressors. The other axis that it acts on is the one that produces epinephrine or adrenaline from the adrenal medulla. And this happens by activation of the sympathetic nervous system, and PACAP has been shown to regulate or turn on this stress axis in response to psychogenic as well as physiological stressors. And PACAP has been shown to act at multiple levels of this pathway, probably high-level regulation from the brain, but also down here in the pathways. So why the heck am I interested then in a stress peptide when it comes to obesity and the metabolic complications of obesity? And that's a really great question. And it really came from some observations that we made during my PhD, actually. So this is actually quite a nice talk for me to pull together because I got to go way back and try to tell a story from the very beginning. So at the time when I was a PhD student, I worked in an endocrine lab that was interested in the evolutionary history of endocrine factors. And so they had identified PACAP as a very highly conserved neuropeptide, but we didn't really know what it was doing. And this was in the late 90s when there was a lot of work going on in sequencing genomes, and new tools were coming available to understand the function of these genes. So we call it the area of functional genomics, and one of those tools were knockout mice. So you could manipulate the genome of a mouse to remove a gene, and then you could see the functional consequences of what happens when you remove that gene. So it's quite a powerful tool to understand gene function. And so that was my project, is we were making a PACAP knockout mouse, and it was me and another graduate student who did that work. And we got quite an interesting phenotype from that little mouse. So quite nicely, because that's what I was really worried about, the mouse actually lived to survive in utero, and they were born, and they were actually born at the same weight and size as their little littermate controls. But as you can see from this picture here, this is a mouse at 14 days postnatally. They started to have this quite severe wasting syndrome, and most of them would die by their second postnatal week. So that's this graph here, the black bar, most of them were dying. But interestingly and rather serendipitously, we discovered that if you raise the temperature of their housing environment, you could actually increase the survival to about 80%, which was staggering with just 3 degrees difference. If we expose these little guys to a cold challenge, they didn't fare as well as their littermate controls. And we also noticed they had reduced amounts of norepinephrine, which is that hormone from the sympathetic nervous system, which regulates thermogenesis. If we looked at the adults, the ones that had survived, there wasn't actually very much striking about them. They did have slightly reduced body weight if they were raised at a colder temperature. Their energy expenditure was about the same as their wild-type littermate controls. We did see a slight increase in activity in the dark phase, and we saw a decrease in the expression of genes that were related to thermogenesis. So that was sort of a summary of the findings. And while this work clearly documented a cold-sensitive phenotype in these animals, we really had no understanding of what was going on. What were the mechanisms? Why was this happening? So now you get a little biology lesson on thermogenesis, just a little diversion, and then we'll come back to the PACAP mice. Oh, I was supposed to ask you guys, where does thermogenesis occur?
Where?
Yeah, where in your body? What happens when you get cold? Shiver. Yeah, Great, so we shiver, acutely we shiver, right? And that's mechanical heat that's generated by fibers rubbing together. What about in the long term? How come, maybe I shouldn't say this, but how come right now -15 doesn't feel that cold compared to like 3 weeks ago, right? So what happens in the long term? How do we adapt to cold? Yeah, brown fat. So our adipose tissue, actually. So we have two mechanisms for thermogenesis as mammals. We have mechanical and also a biochemical mode of thermogenesis. And don't you hate these graphs? The students always hate it when I put up stuff like this, and I don't know how I became a scientist that enjoyed graphs like this. But look at this, it's so great. So if you go into, if you expose an animal to cold, and you put them in the cold, acutely you'll shiver. But you can't maintain that long term because it's very energetically expensive. So over time, if you remain in the cold, you turn on the proteins that are required for this biochemical pathway in adipose tissue, so that after about 3 weeks of cold exposure, you have fully acclimated adipose tissue and you have a minimal amount of shivering. Okay, and so if we do this, please ask questions about animal ethics if you're concerned, but we do expose animals to mice to a 4-degree cold challenge. We do it gradually, but by 3 weeks, they are fully acclimated to cold. If you observe them, they're not shivering any longer, and they have their brown adipose tissue fully turned on, and they're quite happy at 4 degrees. Or I think they're quite happy at 4 degrees. They haven't told. So adipose tissue then is a very important role in thermogenesis. And for those of you who didn't know, you have all sorts of different types of adipose tissue or fat tissue. We have the very classic white adipose tissue that does play a major role in energy storage, where we put our excess calories. Adipose tissue is very specialized for the storage of triglycerides, which is quite interesting because other tissues really don't like having a lot of triglyceride in them, like the liver, right? So, and then we have brown adipose tissue here, which is highly specialized for the heat production. So actually, its main role in the body is energy burning. Yeah.
This slide that we just showed us.
Yeah. Yeah.
Does this happen in humans?
Well, we're learning a little bit about that. We do get, and I'll come on to that, we do get some acclimation. We don't put humans at 4 degrees and see how they fare for 3 weeks, right? They would complain. But we do know that you get an activation of brown adipose tissue after only 5 hours of cold exposure. So you can see the proteins being turned on, and we think the best evidence actually comes from communities of people that are living in, for example, the Arctic, so Inuit populations, where they see, so prior to 2007, you would say, well, we don't have brown fat, only babies have brown fat, or mice, or whatever. And actually, the only human population that was sort of documented to have brown fat long-term as adults was in Inuit populations. But that all changed in 2007, and I'll come on to that in a little bit. So the evidence is pointing to, yes, humans do have adipose tissue, but it's not so much in brown adipose tissue. And I'll just come to this other type of adipose tissue that we have as humans. And other mammals have it as well. So there's another type of adipose tissue that has recently been discovered to be present in white adipose tissue. So I'll just go back. Sorry, being a bit funny. So you have this particular fat that is this picture here. It's from intra-abdominal fat, which is highly specialized for energy storage. We then have other depots of fat, for example, subcutaneous fat, that also will store energy. But if needed, it can turn to a brown-like phenotype and have thermogenic properties. And that previous picture was that type of fat isolated from an animal that had been housed at a warm temperature. And this is taken from an animal after they've been at 4 degrees for 3 weeks. So you can see that there has been a remodeling of the tissue. There has been a change in the morphology. And if you measure the expression of thermogenic proteins in this tissue, they are all elevated. So this can sort of be a backup thermogenesis, because as adult humans, we don't actually have large amounts of brown adipose tissue, but we do have large amounts of this white adipose tissue that can convert to thermogenically active tissue. So that's what we think is happening more in terms of contributions to energy expenditure in humans. So adipose tissue then is a key thermogenic organ in the body, and this all happens, the control all happens at the level of the brain. So the cold signals are sensed by the brain, that message is transmitted via the hypothalamus, and then the stimulus for the thermogenic activation of the tissue occurs by the sympathetic nervous system. And so, does this matter in humans? And that's what I wanted to make sure that people understood, is that it's now understood that humans do have these thermogenic adipose tissues. So it was in 2007 that it was identified that adult humans do in fact possess thermogenic adipocytes that can be recruited in response to cold. We just don't know about the full acclimation process in humans because we don't expose them to a controlled environment where we're putting them, say, at 4 degrees. And interestingly, these are association studies, but the amount of thermogenic adipose tissue and the activity of thermogenic adipose tissue is related to things like age. So as we age, we have less thermogenic adipose tissue, and we have a decreased potential for activating it. And in patients who are overweight or obese, we see lower amounts of thermogenic adipose tissue and an impairment in the ability to activate it. So is this a cause or an effect of obesity? We don't know yet, but we're working towards that in the field. And I think we all are aware of those horrendous weight loss lowering drugs that stimulated thermogenesis, and these were done historically through an indiscriminate activation of the sympathetic nervous system. So you were seeing the effects, probably weight loss through activation of thermogenesis, but of course you were having all sorts of other side effects on the cardiovascular system. So again, this has been a bit of an explosion of research for the endocrine field, where we're really trying to understand, where did these beige adipocytes come from? How are they recruited? What turns them on? What pathways are controlling them? And so that's where my NSERC grant has been founded, in just really trying to understand some of the factors that are regulating cells. Okay, so back to PACAP. So the fact that the PACAP knockout mice have this cold-sensitive phenotype, we had a hypothesis that perhaps, given its role in the stress response, that in response to environmental stressors, perhaps PACAP was involved in regulating adaptive thermogenesis by the sympathetic nervous system. And so this is the part of the energy balance equation that we have been looking at. And so the first thing we did shortly after, maybe a few years after I arrived at UNBC, I had the PACAP knockout mice brought here to UNBC, and we have a colony here in the Northern Health Sciences Center that we work with. And so one of the first things we decided to do was do a protocol that was developed in Sweden by Dr. Barbara Cannon. How am I doing for time? Does anybody know the time? 1:40. Okay, good. All right, I should be done within the hour. So this was a protocol developed by Barbara Cannon in Sweden. And what they do is they have mice that are anesthetized, and then they put them in an indirect calorimeter, and that's a way to measure oxygen consumption. And so then they give them an injection of norepinephrine, and we watch the changes in oxygen consumption. And so if you take an animal that's been raised at 24 or 30 degrees, when you give that, because they're anesthetized, if you give them the norepinephrine, there's very little change in the metabolic rate. But if you do it into an animal that has been housed for 3 to 4 weeks in cold first, you get this very dramatic increase in oxygen consumption, which is actually thought to be the activation of that thermogenic adipocytes, because it's very, very energetically expensive. And so we did this in our wild type animals. So these are just normal mice. And we did this experiment at 24 degrees and then at 4 degrees. And we see this increase, as we should, in energy expenditure in animals that have been raised at 4 degrees. When we do it in the PACAP knockout mice, you can see that we get no induction here at 4 degrees, identifying that they aren't able to induce that adaptive thermogenesis in the cold. We then also noted that this is a complicated slide, but what it means is that during this experiment, we measure the nutrients that they're using, and they should be using lipids to drive that process, because fatty acids drive that process of thermogenesis. So all the other animals did that. They come to 0.7, which means that they're using lipids. But you can see that the knockout mice don't seem to be able to use their lipids. They move towards carbohydrates. So we could see that they were using fuel differently, and it seemed to impair their ability to induce adaptive thermogenesis. Surprisingly, their brown fat looks quite similar, but we see a complete impairment in their ability to turn on these thermogenic proteins. So this is a protein called uncoupling protein 1. They're not able to turn that on. And we see downregulation of some of the genes involved in the pathway turning on thermogenesis. And so based on these results, then, we felt that PACAP was somehow involved in the regulation of thermogenesis. And in the meantime, many people were searching out other hormones that are involved in this process as well. So now we know that there's tens of hormones that are involved in this thermogenic process, and we think PACAP needs to be added to this list. So I'm now just going to buzz through some of the current work that we're working on without going into a presentation of data. I wasn't sure how much time I would have, and I didn't know how much people wanted to know, honestly, about the details of all of this. But I'll just talk about some of the key research questions, and then I'm going to go through one little project that we've been working on that's just recently. So this is some current work that actually Megan Forrester, who was an undergraduate student, she's now in vet school, but she really would say to me all the time, but if they can induce adaptive thermogenesis, how are they living in the cold? I don't understand. It just bothered her. And so I said, okay, well, let's find out, how are they doing this? Because the PACAP null mice, if we take them and very gradually introduce them to this 4-degree cold exposure, they can survive. So she said, why? How are they surviving? So she took groups of animals. Have I got this labeled right? Yeah. She's taken groups of animals. She's used the wild type mice compared to the genetic knockouts and put them in warm and in cold. And she's asking questions about, can they prolong their shivering? So remember I showed you shivering should turn off as the adaptive thermogenesis goes on. So she wanted to know, can they prolong their shivering? So what she's been doing is she did some observational studies looking at shivering behavior. She didn't actually record any, or she was just checking them once a day, but she didn't see any changes in shivering behavior. But now we've collected the skeletal muscle and looking at the molecular structure of the skeletal muscle, because it's almost like endurance training. So if you shiver long term, you get more oxidative fibers. And so Ekaterina Filatov, a student in my lab here, she's been using immunochemistry to look at the fiber type switch that would happen in the muscle. And it looks like the PACAP null mice have more endurance trained muscle. So they may be shivering to maintain themselves in the cold. The other thing that she was quite curious about was this browning process of white adipose tissue. Is it different between the knockout animals and the wild type animals? What we know so far since 2007 that the regulatory pathways going to brown adipose tissue and the white or the beige adipocytes and the white adipose tissue is similar, but nobody really knows yet about that general physiology. So we're having a look there. So these three are mostly working on this, and we're almost ready to put a paper out on this, looking at the muscle in these animals. So really, my great interest in this is trying to understand where PACAP is acting. And I actually think it's acting at the level of the hypothalamus. I think I talked to you at the start and said it's sort of a master regulator of this stress response. PACAP is highly expressed in the hypothalamus, in centers involved in appetite regulation and energy expenditure. And so it wouldn't surprise me at all if we find that PACAP is expressed in the hypothalamus is actually controlling these downstream pathways. So we have a project right now that is kind of taken 2 master students to move through, where we're going to take our knockout mice and reintroduce PACAP just into the hypothalamus and see if we can rescue that phenotype. But that's taking us a little while because it requires the generation of novel animal models and things like that. So we have these 2 master students that are working on it and has been very much supported by Landon here in the lab. The other area is that we probably shouldn't overlook that there could be a direct role of the hormone on the adipose tissue itself. And this is one thing that Damon came up that he was really interested in looking at. So this is Damon Klein, master student in my lab who'll be finishing this year. And he said, yeah, but we don't even know if adipocytes express PACAP receptors. Remember, the hormone has to have the receptor. So he's really taken this on in the lab. He's asked if PACAP could be acting here. So I'll just show you a little bit of data that he's just presented recently at a conference and the work that we're following up with there. So again, remember, you have to have this receptor if your hormone is going to act on that tissue. So that's where Damon started. He asked, and so just to make things complicated, I told you PACAP is my nemesis. It has 3 receptors that it binds to, not just one. And this one here has like 11 splice variants. So it's a very complex receptor system. And so he just really started with quite a simple question, but never simple in PACAP, is are PACAP and PACAP receptors expressed in adipose tissue? And we thought, well, while we're doing it, let's see if their expression also changes with environmental temperature. So he took his mice and he had some at 30 degrees and some at 4 degrees, and he said that but they still maintain their body temperature at 37. They were exposed for 3.5 weeks. He evaluated his model. Did I have a good cold exposure? And then he looked for receptor expression using real-time PCR and RNA sequencing data or genomics. So this was one of the ways he validated his model. You can see that the adipose tissue looks very different in animals that are raised at 30 degrees, where they don't need that thermogenesis at all, versus when they're in the cold, where you see the brown gets browner and the subcutaneous fat gets more beige, and this one stays quite similar. So what did he find? I'm just showing you the expression data here, and I'll tell you a little bit about what they're finding functionally. So the first thing, this was part of his model validation, and you can see in his cold animals, he sees this dramatic increase in this thermogenic protein and others. And then he looked for the receptors in PACAP. So he showed that the adipocytes themselves don't make PACAP. So PACAP is probably coming from elsewhere, but they do in fact express these 3 receptors within the whole tissue. Then we wanted to say, yeah, but adipose tissue is made up of all sorts of different cells. So let's see if they're actually expressed in the adipocytes themselves. So he developed a primary cell culture model where he can take the tissue, isolate the cells, and then check for the expression. And he sees the PAC1 receptor and the VPAC1 receptor in the adipocytes themselves. And what we found was that we don't actually see expression levels increasing that much with a cold challenge, or they do a little bit, but this one goes down, but we don't see much change with cold exposure. In the white adipose tissue, remember we took 2 types, one that browns and one that does not. We know he got the right depot because look at this induction of a thermogenic protein with cold. And he sees 2 of the receptors expressed, but again, no PACAP. And again, he sees them expressed in the adipocytes. And we do what's interesting here is we do see some regulation that is similar to one of the lipogenic genes, which I'll come on to in a second. And then in this fat that doesn't brown, same thing, he sees the different expression pattern, and this is what I'm referring to, is it goes in the same direction as this species cell. So actually this data, along with data that we've had from the oxygen consumption thing, suggests that maybe PACAP actually involved in making that fuel available through lipolysis. And so he's been doing some assays with his isolated cells to say, okay, now I know the receptors are here. What are you doing in these cells? So he's taking PACAP that he's purchased. We can buy synthetic PACAP. He's adding it to those cells and then looking at the functional effects on the adipocytes. And that work will be ready for the undergrad session in the research week. So I thought I better not steal their thunder. So this work is being done by Damon. And so this is the work here being done by Simon as an undergraduate student. And then Colin Appleby is a student that came to us this summer who now is saying, so I want to look in the whole animal. Do they have any changes in lipid metabolism that might be contributing to this impairment in thermogenesis? Okay, so I think that's where I'm going to end. So I hope I've been able to convince you, perhaps, that PACAP may be a novel hormone regulating thermogenesis and therefore energy balance. And this kind of work contributes mechanistic puzzle pieces to the big picture. So I'm not finding a way to isolate insulin and run it down to the clinic. Most of the translation of biomedical knowledge to the clinical field takes years, if not decades. But we do feel that we're making small contributions to the puzzle pieces to understand this energy balance equation. And in our case, perhaps how stress impacts this energy balance equation, which we see in the community and in the clinical literature. And then, of course, from a biomedical perspective, there's always this idea that your molecules or your signaling pathways that you're identifying could lead to therapeutics. I don't see that just in the context of pharmacological development, but also perhaps in understanding, for example, why certain lifestyle changes will help to modify energy balance. But if people want to ask me questions about pharmacological development for anti-obesity therapies, I definitely have an opinion on it, and I'm happy to discuss it. So I'll just finish there by just thanking the lab again, and of course a thank you to Lydia Truck and Sian and Natalie for the care and attention that they provide here at UNBC to our animals. Thank you.
So Sarah, thank you so much. It's so impressive to have these opportunities to let you dig into, well, I find it impressive. I find it a real treat to actually be walked through what you're doing and how you're doing it. I've certainly got a few questions up my sleeve, but I'd really like to open it up to anybody else who would like to dive in onto any of the history or the detail or the graphs.
Yeah, I hope it was interesting to people. I was really struggled with what level to pitch it at. So yeah.
Chelsea, where to go?
I was curious if you noticed in the polls, is either the PACAP or the wild type, are they more active?
Yeah, so we did that graph way back where we actually had them in a metabolic chamber for a 72-hour period where it was quite a sophisticated system where they had laser beams that we did see increased activity in the dark phase, so when they're active. Megan did a study where she just sort of popped in. She'd taken an animal behavior course where she had recorded activity, and just her once a day pop in didn't reveal any differences in activity, but I don't think it's sensitive enough because the animals are active at night. We have also seen that when we put them in an indirect calorimeter, when they're not anesthetized, we have slightly higher rates of energy expenditure, which suggests they might be slightly more active. And so we definitely think activity is perhaps a way that they're compensating to keep warm. And we've suggested that sort of quite a while ago, but for sure, because also we see in the adults that survive, if you raise them long term at 21 degrees, they have a lower body weight. And so we think that they're probably moving to compensate for the lack of adaptive thermogenesis. Yeah, we need little Fitbits. If you can figure that out, that would be great. Yeah, there are systems called metabolic caging systems. I just don't have them here at UNBC. And because we've done the experiment before, I haven't shipped a cohort down. But yeah.
Other questions?
You mentioned
Judy. Judy's my part-time lab manager. Yeah.
I was curious to know, your temperature difference. What about emotional and psychological stress?
Yeah, I haven't gotten into that, Judy. I don't want to subject my little mouse to psychological stress. You know what? It's just not something that we've done. There's quite a diverse field in PACAP, and there's actually quite a body of literature on anxiety in the PACAP male mice. The other interesting thing, really probably the most relevant human data for the hormone PACAP, is in the field of PTSD. So they've identified a gene variant that, or a single nucleotide polymorphism that in the PACAP gene that predisposes women to more severe presentation of PTSD. So, and they think that they've worked out the mechanism to be via the estrogen receptor in, I can't remember the region of the brain, but it seems that PACAP is involved in that regulation of stress response. And when there's an impairment in PACAP function, that you can see presentations of stress disorders. So there is quite a body on anxiety and also PTSD, but probably not something we'll get into here. It's just not my area of expertise at all. Yeah, but it is an area.
I was going to ask about, is there reproductive connections or a link with estrogen? If temperature changes that population and fat is metabolically active for estrogen, those sorts of things, infertility perhaps.
Yeah, so So we don't breed the knockout animals. We always breed heterozygotes because that's sort of the gold standard for maintaining an animal colony. But there was a woman after I left my lab at UVic who did a study on the reproductive success of the knockout animals and saw impairments in the female knockout animals. But they're a tricky bunch to work with because their pups are also dying. So it's just difficult to sort of catch when that occurs. So actually I should dig that paper out. It's from years ago and I can't remember the findings other than there was some sort of fertility deficit in the female. But for sure, so this peptide is kind of everywhere. And that's the challenge of working with it really, is that there's a lot of secondary factors. So we've tried to hone in on experiments, and that's why we're doing some of these manipulations in like reintroducing the gene in particular areas, because we think that we need to dig a bit deeper to get to the actual location of where we're seeing the problem.
Sarah, I've got two related questions.
Okay.
And they both relate to, one of them's just a personal curiosity, but they both relate to doing this research here in Prince George, and that's in our territory. But one of the things, I love the fact that you gave us those little micro lessons on really core principles, and the thermogenesis stuff is for things I haven't thought about for years. And so this is really a personal question around when I was seeing that initial graph of the induction of the adipose tissue in the mice, and then we already had the question about what's happening in humans, and you talked about the shift in understanding of thermogenesis in relation to the beige adipose tissue, which I didn't even know about, which is interesting.
More of that.
But one of the things that I found myself wondering about, because quite literally, this is the personal question, when I moved to Prince George as somebody who had never experienced cold temperatures like these for a prolonged period of time, and I know that this is not an unusual anecdote, I just had the overwhelming desire to eat fatty sausages. And it wasn't just fatty sausages, but it was like, I need animal fat. Like, it was just a sort of totally, it was a very, and I like food, clearly, but it was this sort of sense that something has happened in terms of my response to this environment. And so part of the question is the activation of the beige adipose tissues and that browning of the beige tissues, is that an energy intensive thing that actually requires you to want to eat animal fat?
And probably. And so we're always, I think when you think about human biology, we're often thinking of things in a backwards way.
Sure.
So I'm going to digress, but I'm coming back.
I'm glad.
So remember when I showed you the leptin thing and I said it sort of happens this way? When you have high leptin, that turns off feeding. Actually, what really happens, we can't use leptin as an anti-obesity therapy, because what happens when individuals become overweight is they become resistant to leptin, which makes perfect sense from in a biological context, because most animals don't live in a space with nutrient excess. So actually, if you encounter long periods of nutrient excess in the wild, you'd be like, awesome, let's utilize this energy. We'll store it in our adipose tissue, because there's going to be some times of famine coming along. Do you get what I mean? And so I think we're often thinking about these things in a backwards way in terms of how things happen with physiology. So yes, I think that an animal is always trying to, or in biology, I should say, we're trying to maintain this energy balance. So as somebody coming into a cold environment, having to spend that extra energy at the expense of your metabolic health, you need to overcome that extra use of energy. And the way we do that to rebalance the equation, if you're burning a lot, is you're going to eat a lot to balance that back. And I have just experienced that. I've become a new farmer, everybody. I have cows. And our cows absolutely went crazy at 0 degrees, but about 4 days before the temperature dropped, they ate probably triple. And the temperature was 0, but it's like they knew this cold spell was coming. Fascinating to me. But yes, it's natural, I think, to rebalance that equation. And that's one of the things that I think actually PACAP may be involved in. And maybe by the time I leave UNBC, I will have tried to prove it at the level of the hypothalamus. But I think PACAP is setting the set point in the hypothalamus. We've had little bits of evidence that suggest it may be that the set point is off in our knockout mice. So 24 degrees is actually feels colder to them than it should. And I'll use the example of the muscle. Chelsea, you might be kind of interested in this. If we look at the muscle from the mice that are raised at 30 degrees, it looks like muscle from the wild types that are in the cold, suggesting they're kind of like cold at 30, but they shouldn't. It should be thermoneutrality for them. And so we're wondering if this idea of set point, and I think that's exactly what happened to you. Your set point was off. And so you weren't used to having to expend that much energy on maintaining your body temperature. And so then you're increasing your appetite. But I don't like to say too much about your personal situation, but I feel like that might have been what was happening. Yeah.
So I've still got 2 more questions.
And can I say one more thing about set point? And that's the other reason why when you have individuals who try to diet, why it's actually you're having to overcome physiology when you lose weight because you have a set point that your body is trying to fight. And so it's a very well-known phenomenon that goes on for the literature I was reading prior to giving this talk was for about a year, that your body will fight that physiological set point. So actually the drive to eat is probably more overwhelming than someone who is not trying to lose weight because you are actually readjusting that set point at the level of the hypothalamus. So that's a clinical phenomenon that you get this rebound of weight gain when people have lost weight through dieting and exercise, and that's partly to do with this energy balance set point. Yeah. So it's a challenge, a real challenge, and not just because all of us, it's a challenge from our outside factors, but it's also a challenge because we have our inside factors working against us. Sorry, I interrupted your second question.
The second question I had is closely related, and it's to do, not surprisingly, I'm interested in this dynamic between the outside and the inside factors.
Yes.
But I'm curious about, given some of the, again, there's a lot of risks in extrapolating to humans from what you're doing.
Yeah, for sure.
Yeah. But is there, it's just a curiosity, is there a particular type of interest in some of this research into thermogenesis? You're looking at a particular hormone, but is there a particular interest in cold places?
Yeah,
and
I think
an area that is, you mentioned the Arctic, which is certainly obviously one area where in this way in which it must have changed the thinking to understand these dysphagia adipose since 2007 and so on. But is the notion of the heat and thermogenesis dynamic and people and living in cold temperatures?
Yeah, I mean, I don't think that it's coincidence that the groups that are the experts in the world on thermogenesis are from Sweden. That's where they're from. Yeah. So I think there's sort of an interest in cold temperatures and thermogenesis for sure, and physiology in general. There is a group, a group's probably leading actually, or a leader in understanding these processes in humans in Quebec, in Sherbrooke, so north. Andre Carpenter is doing all of this work, but in humans. And so he's trying to translate what is being found in the lab to human models. He has a very, very sophisticated metabolic lab where he does expose humans to short cold exposures, but then also does pharmacological exposures to replicate long-term cold exposure. I don't know what he compensates his human subjects with. I'm not quite sure how they get these people to do these studies, but they're very intense. And so he's really moving the field forward in terms of that translation. What does this mean in the context of human biology? Yeah. And so, it's hard to speculate to humans, but I think there's a natural curiosity for our environment and how we're influenced by our environment. And I know the work that I'm doing is not necessarily specifically northern, but I think being in a northern environment gives you a bit of interest in terms of these kinds of processes. And I don't know if that's why we've gravitated to thermogenesis in the bigger picture of the energy balance equation rather than appetite, but for me it's of interest. Yeah.
Room for other queries? We're in the final wrap-up zone here. So it's actually a direct rip of what you just said, which is I'm really glad actually that you chose to focus on the biomedical piece on that section, because it's fascinating work, and it's really neat to also meet your lab. We've been hearing about, they're just And although you were more talking about this end of things, I have heard you describe previously, and you've alluded to it today, this question of how your research portfolio across that spread has been influenced by the kind of environment that you're here at in Northern BC, not just the cold thermogenesis theme, but I wonder if, as you're finishing up, if there's any examples that you could give about how your work may be evolving or have evolved in different directions because of that cross-disciplinary interaction.
Yeah, I think that for sure the clinical project that I started with Candida Graham is not something that I would have embarked on had I, say, been doing this work in a large center in, say, a diabetes or obesity field. And I feel like probably just trying to make, I think one of the things that I focused on being here is just trying to always ensure that biomedical research is part of the health research conversation, because I feel it's really important that we need to have the spectrum of health research to be truly successful and world-class as health researchers. So I'm not sure about all the potential collaborations that will happen from it, but that's where I've tried to dedicate some of my time, whether it be engaging with Northern Health or trying to sit on like the Northern Node meetings or join the HRI, is just try to keep that perspective. And I don't know, hopefully I'll feel like it was important for me to be here the day that I leave.
Well, we already did.
But I think it is in terms of, there's lots of ways that we contribute to the community that we work in, like training trainees or teaching physiology to the medical students and things like that. And I think just even like Chow's here today, but just building a cohort of people who are involved in biomedical sciences. You can't do this kind of work alone. The equipment and the reagents that we use are very expensive. And so we have an example in the Northern Health Sciences Center where we have sort of now like a group, like a shared equipment that has allowed Chow to diversify his research program to be directly Northern. So I think it's building that research capacity is really important to, again, the institution. And if I can contribute in that way, then I feel like I have made already contributions that way. So, we'll see.
Thank you. Well, I think it's really important to remind us about the way in which that collaboration is happening among different researchers and these other potentials. And I'm excited to be able to speak slightly more informedly about some of the work that you're doing, or my interpretation of the beige fat.
And celebrate your beige fat.
Split that into dinner time conversations. So I'm not seeing any final burning questions, but I am going to return us to the invitation that if there is anybody that actually does feel like they might want to be the March 12th speaker, or if you know of finishing students or somebody that would, or a couple of folks that would have to be a really good opportunity, we're just having had a change in plans for one of our speakers. We've got a space. But it is my great pleasure to say thank you, Sarah. That was fascinating. Thank you. And thank you for doing a great talk. I think you've seen one of these. It was great. I really enjoyed it. I really enjoyed it. I know I'm doing it. Yeah, it was great. I found it fascinating. I'm glad.